All right, so welcome back. Today we're going to be focusing on topic F, which is all about classification. We're going to be looking at prokaryotic and eukaryotic classification. And so that's mainly organisms that we know prokaryotes don't contain a nucleus, eukaryotes do, correct?
Correct. That's how we're going to separate them out, at least for this part. So the focus is going to be the main ideas on how to classify things. That's where we're going to start off. So the rules. And then little by little, we're going to look at examples and what are the characteristics behind them. So if you see the list of SLOs, you'll notice that SLOs C through G all look pretty much identical. The only difference that happens between them is which type of organism we're going to be focusing on. So for the main idea is what is the organism, how do we classify it, and what are the properties associated with them.
All right, so let's start breaking some things down. So we've got taxonomy, classification, identification, and nomenclature. We talked about nomenclature a little bit, how we name things. Do you want to break down some of these other?
Right, so nomenclature was about naming. That was pretty much the beginning behind it. The problem with that is that just is the name. However, by properly naming them using the genus and species that we had talked about before, that allows us to create categories, or what we call bins of classification. In other words, we're sorting them out into things we can actually agree or disagree in terms of their similarities, how they work, their functions, their needs, and so on and so forth. But there is a better way to do so. Classification is very general. It's kind of like sorting things out just based on what we think is funny or important, or not even anything that makes any sense sometimes. It could be by color, it could be by size, and those are not quite useful in science. But in science, we do have a better way to do so, and that's called taxonomy. So it's the science of classification. And we're sorting things out here more or less based on their DNA, possibly some of their functions, possibly some of their organelles, how do they move. And these are all based on a hierarchy, meaning that the more complex they get or the more related they get, the higher the taxonomy gets. And the purpose behind doing so, aside from just the nomenclature and the name, is so that we can identify them. If we know what properties they have, we can properly name them and then use them specifically for medical treatment. So the more we know about things being related, if two particular organisms are very similar, that means that more likely a disease will hurt them both roughly the same. Or also in terms of treatment, if they're similar, it's most likely that if a treatment works well on one, it will also work on the other. So that means that what we're going to be looking at is the field of taxonomy, and that means also that we're sorting out all of these particular germs, pathogens, whatever you want to call them, into categories that we call taxa, or singular is taxon, that allows us to sort them out in this hierarchical organization, meaning the higher they get, the more related they are. And this is largely based on DNA and their phylogenetic or evolutionary relationship.
All right, so getting into taxonomy, we're looking at what you were saying earlier, that hierarchy. So kingdom is like zoomed all the way out, basically, and we can jump into phylum, class, order, family, all the way down to specific epithet or the species, right? And that's kind of as specific as we can get, but we can divide each of these by these things that we see listed over on the left, right? Like I was mentioning earlier, prokaryotes don't have nucleus, eukaryotes do, things like that.
Right, and so once you start getting higher and higher on that system of taxonomy, some of these properties start disappearing or reappearing together. So certain organisms, depending on how they reproduce or divide, will be categorized together, whether they move, their shapes, and like you said, ultimately way at the top, we end up having several different kingdoms that divide organisms into eukaryotes and prokaryotes. The problem with that is that that is no longer the case. What we used to learn as the five kingdoms of life has now kind of disappeared, because we now know that some of those relationships are a little inaccurate. Now we know that, for example, what used to be kingdom Monera has disappeared and become Archaea and bacteria as separate categories called domains. And then everything else, putting your plants, your mushrooms, and everything else, fall into one giant category called eukaryotes or eukaryota. That's really where they fall. So when we look at this in terms of a phylogenetic tree, all the organisms are still related ultimately into what we call a last universal common ancestor, or LUCA, or LUCA is the other term we use. And then from there, they start branching off millions of years ago, around 4 billion years or so. And then we get our bacteria in one group, we get our archaea in the other group, and then everything else, all the eukaryotes, the ones that do have a nucleus, get separated out. However, there are still organisms out there, for example, things like viruses, that we still haven't been able to classify fully. So that means we're still missing some information, or we haven't figured out what properties to properly use between them.
All right, so this is getting into some of the ways that we can differentiate. We're going to be looking at shapes, and we talked a little bit about this, and we're familiar with like cocci, bacilli, all that stuff. We're going to do a quick review of that.
Right. So if you recall, we separated just organisms based on their shape under the microscope. And so, like you mentioned, some of them can be spherical, some of them can be rod-like, but there are other shapes as well. And we'll normally focus on the easiest one since that's what we look under the scope, but there's plenty of variations not only into the individual shape of the cell, but also their arrangement, whether they're 1 by 1, 2 by 2, long chains, groups of them, and that's kind of what we have listed on the side. Similarly to that, we also look at their shapes when they're growing on a plate, and so that's their colony. And when we looked at them, we classify them based on how they grow, their look, their three-dimensional arrangement behind them. Some of them pretty much based on the edge of their colony, if it was kind of nice and sharp, or it had some ridges, or it was what we call filamentous, meaning it started having these strands sticking out. And then how they grew overall on the plate, if they were just by themselves, or if they clustered all together and they had a little bit more of a raised format, or they kind of started stringing together. All of those give particular shapes too. And those are other forms of those types of classification. Now, aside from that, we can also classify them based on their color, based on their shine, based on how their consistency is, especially when you start inoculating samples. So some of them kind of look kind of dry, some of them break off, some of them are kind of sticky, and all of those have their own categories. So those are all ways to classify colonies on a plate. But we can also look at how they divide. And the classic way of looking at this is a type of reproduction that most people look in A&P, and it's talking about your classic reproduction based on sexual reproduction. Nothing that we're going to cover in here, that's not our topic, but when we focus on mitosis, that's usually the one that's very similar to relate to most bacterial growth. In particular, binary fission, in which you usually take one cell and it divides to 2, 2 to 4, and so on and so forth. A variation to that is called budding, in which instead of having a symmetrical growing, they end up having one kind of popping off the other, like the term budding in itself. And there's some even weird enough that they kind of refer to as snapping or hinge division, in which they start dividing symmetrically, they start extending, but they never separate fully. So they end up having kind of this little hinge-like look to them. And so the mitotic similarity to them, this is the so-called binary fission that you're seeing on the right-hand side. The budding version is what you're seeing on the left-hand side. And eventually, once we start looking under the scope, we can actually see things called palisades that give us this snapping division or hinge division also. In topic E, we also looked at what things use to divide. So in terms of their energy source and where they're obtaining their carbon. So that's what this table is summarizing here, dividing organisms into heterotrophs and chemotrophs and so on and so forth. But then we also needed to look at how we can classify, for example, bacteria based on certain physical traits. The main one that we always use in terms of these organisms is the wall, and this is what we're doing in the lab by separating gram-positives and gram-negatives based on how much of this extra little wall do they contain, the so-called peptidoglycan, and whether or not they possess a secondary membrane. So these are all examples on how we sort things out based simply on external factors or just shapes or arrangements. Now, when we start doing this in the lab, we look at specifically how to stain them, but in reality, when we do this for classification, we're looking at their content. How much do they have? What are their linkages in terms of chemistry? How do they react to certain treatments? That's what we're going to actually use for classification itself later on. And then lastly, the staining is one of the last forms that we can use, whether gram-positive or negative or blue or red, as a way to dictate what type of organism it really is. Now, There are other ways to do so, and then aside from just doing little stains and looking at morphology under the microscope, there are biochemical tests that we're going to do that we can look at what do they consume, how much energy do they produce, and that's what we cover in the labs about 19 through 38 by testing things changing in color. But then we can also use their reaction to the immune system, in other words, how they produce antibodies when they infect certain hosts, so that's a field called serology. In certain types of organisms that infect just bacteria, this is called phage typing, in which viruses that infect bacteria can tell us what type of organism they're looking at. So that's the term phage that we'll look at later. And then lastly, the most simplistic of them all, although a little bit more complex to do in terms of the lab, is looking strictly at their genome, looking at their DNA itself. So we will actually end up doing this in the lab aside from anywhere else. But these are all the tests that we look at when we're trying to classify things in this subject. So what we want to do is, based on kind of those descriptions that we just gave you, is how do we classify things, how do we give them names, how do we identify them, and we're going to start with the first set of prokaryotes, which are going to be our archaeans, and then followed by bacteria. So we start off with our prokaryotes.
Right.
So unfortunately, when we cover prokaryotes, currently we've agreed that there's about 27 different phyla that fall under bacteria. Problem is, we're still finding new ones, we're still discovering new types of organisms, and the estimate says that it could rise to about close to 300 of them. So clearly we cannot cover this in all classes, so what ends up happening is we end up highlighting the ones we're interested in. And so we're going to focus on the first group, which is going to be just the archaeans, and those are a little bit smaller in terms of the groups since they're kind of limited in terms of their presence across the planet. So our archaeans, in this case, some of the unique features that you can see here on the slide is they do not have a cell wall. In other words, there is no peptidoglycan. Now, their membrane does have some almost similar or kind of wannabe peptidoglycan, but they don't have a true wall. So in this case, they're actually more closely related to humans because we don't have walls, as opposed to bacteria. They also divide very similarly to all other bacteria by binary fission or by budding, and they pretty much have the similar shapes. So they look like little rods, like little spheres, and so on and so forth. But the unique feature on how we know how to classify them immediately is, as of this moment, there are no archaeans that we know that cause disease. And this is based on where do they live, specifically their environment or their habitat. But the currently recognized systems, and this goes beyond the textbook currently, is that there are 4 phyla that are recognized. So these are the highest categories that we can get before we get into our kingdom or domain now called Archaea. And so these are the Crina-Archaeota, Uri-Archaeota, Cori-Archaeota. Sometimes that Y can be eliminated, so it's called Cora-Archaeota, and Nano-Archaeota, which is the most recent taxonomy that has been brought up into our microbiology. Now, out of those categories, 3 of them, Crina, Uri, and Nano-Archaeota, are usually referred to as Extremophiles, whereas the Cori-Archaeota, in this case, is referred to a specific group called the Methanogens. So we'll look at them one by one in a moment. So the word extremophile should give you kind of a reminder of what things look like and what do they do. The word extreme kind of gives that hint. This is not an official classification. It's just a category that we use to describe things. And what it really means is the property. They survive in extreme conditions. But what does that mean to you and I? Is that they survive in extreme conditions based on us. Not because they happen to be in the place, is that we don't like that place. So this is a very human category, not necessarily simply because where they live. And so these are based on how high the temperature or how low the temperature can get. Same thing applies to pH or in terms of their osmolarity or the salt present outside of them. And out of those, we have created 2 smaller categories called the Thermophiles, the ones that prefer temperature. And then we have the Halophiles that can vary between salinity or pH. So under there, we're going to have our Thermophiles, the ones that prefer a hotter temperature than usually us, the ones we don't like. And that's usually somewhere above 45 degrees Celsius. Remember that humans are about 37 degrees or so. These ones like it about 7 degrees to 8 degrees hotter. And then we have hyperthermophiles they literally double in our temperature. So that means places that are near boiling that you and I cannot survive in, but they thrive in these environments. And so most of those are under the Crene Archaeota and Archaeota phyla. And so one of the examples that I'm showing you over here is an organism called Geogemma, which survives at 130 degrees Celsius, and then Pyrodictium, which is the one that you're seeing on the bottom. Now, where do you find these guys in such temperatures? Probably the easiest example that most people can relate to are geysers and sulfur vents. So anytime you travel around and you see places that are kind of bubbling and exploding and have water boiling out of everywhere, that's usually where you find these.
So somewhere along the line of like their evolution, they just developed the ability, or weren't taken out of existence because they had the ability to just survive in these conditions. Is it their membrane that just doesn't break down in these extreme conditions?
It's actually quite the contrary, believe it or not. It's very interesting. People just assume they became this. Actually, they already existed this way, and as the environment changed, we changed. They remained the same. Okay. Reminding that the planet was already a giant, ball of heat and a giant mess, and they survived there. As it started cooling down and the environment changed, other organisms arose and became more adaptable to cooler environments and less violent environments. But these guys were already that way. So they didn't become it, they were already that way. So this is something important to understand in terms of evolution. Things don't become based on evolution, they're already there. Mm-hmm. And as things kind of, or so-called evolve, certain changes in their environment allow them to thrive. And the more they thrive, the more they can reproduce.
Got it.
So these guys have been reproducing in that area without any issue. Us, on the other hand, as we reproduce in other environments, we became more adaptable to cooler environments and probably more accessible food than these guys have. Mm-hmm. Now, there is a group called the Halophiles, still part of the Extremophiles, simply because they prefer higher amounts of osmolarity, typically salt, but usually that involves much higher than our classic 0.9% that we're used to in terms of our cells. Most of these organisms are classified under the phylum Archaeota, and the example that you're seeing on the right-hand side is an organism called Halobacterium salinarium, which its name should kind of give it away. The term halo gives you that part, and the term salinarium, also salty, gives you that quick indication. And so these organisms prefer pretty much ocean water or above, so pretty salty environments themselves. Now, the rule is roughly above 9% salt. So in this case, if you want to think about humans, we're about 0.9%. That means 10 times more than what we prefer is already. And that's usually the minimum to classify them. Most organisms under this category prefer somewhere around 20% or so. So that's quite a bit. Now, as you were asking earlier, it wasn't the evolutionary part that made them become, but they do possess organelles, especially part of their membranes in terms of what they've created outside, like a cell wall or so, not that these possess official ones, is that it allows them to withstand high amounts of salt without bursting. Mm-hmm. So remember when we talked about molecular transport, the water flowing in and out through osmosis can blow up the cells or shrivel them. Yeah. Here, because they have specialized membranes, they can actually survive in these environments a little bit better.
So they'll hold on to water better than any human cell.
Right. So we go in that water, we end up shriveling up.
Yeah.
But these guys thrive in this particular environment. Now, the other unique feature that happens to them is because they're usually out in large bodies of water that contain high salt, is that they're usually under the sun a lot. And so that means they're beaten down by lots of UV rays. And just like us, UV usually causes harm. Mm-hmm. And so the way they protect themselves is producing pigment. They color themselves, if you will, by releasing certain pigments that to our eyes look usually in the orange, red, pinkish kind of ranges. And so most of the time when you see them grow in high amounts, it'll change the color of the water to have these deeper kind of red tinges or hues that look dramatically different than other environments. Now, the last group that we have under Archaeans are the Methanogens, and the name should also kind of give it away. The methane part and the gen part saying that it produces. Mm-hmm. And so all of these take inorganic substances and some organic substances and create literally methane. Now, what's the big deal behind this is that for us, a lot of the organic byproducts that we produce, a lot of the waste products that we're doing, things like CO2, they will take it and recycle it. So when we talked about topic one, we talked about how to exploit organisms and they can provide us good uses in the environment. So biorecycling, for example, these guys are part of that group. And so they'll take certain inorganic substances or wastes and actually turn them into something useful. In this case, methane gas, which we can use it as fuel. Yeah. So this is a natural gas version of that.
So are these the ones, are these found in our digestive tract, these methanogens?
Specifically in animals like ruminants, so cows, goats, and things like that usually have that. Okay. And so it helps them process certain parts of their food. Humans, thankfully, don't have this, but things like ants do have them as well. And so they do survive in their gastrointestinal tract, and they help them process certain things that humans at this point in time cannot do. So this is why when you see them chewing on grass, we normally can't do that. The high amount of fiber content behind that is not pretty much useful to us, but they can actually do so. And those leftovers, the methanogens, actually take over and process it and turn it into methane. And so this is why people, when talking about farming industries, they talk about the excess production of methane that usually ends up being blamed for the ozone issues and specifically the greenhouse effect that most people refer to.